Hello everyone. On behalf of all co-authors of this publication, I will briefly run you through our study, risk and protective factors for infection, severe disease, and mortality in epidemic respiratory viruses. My name is Dr. Zheng from the first affiliated hospital, Zhejiang University School of Medicine. Why we perform this review? As we all know, after the COVID-19 pandemic, we have seen a concerning resurgence of respiratory viruses, including influenza, RSV, rhinovirus. This is not simply a return to pre-pandemic level. The epidemiology has changed. Why this is happening? There are several reasons. It may be related to the relaxed MPI, and it may also related to the herd immunity waning. And some concepts like immune debt and immune imprinting or immune safety has been raised. So in this review, we systematically analyzed 9 respiratory viruses to identify both threat and virus-specific risk and protective factors across 4 key outcomes: infection, hospitalization, severe illness, and mortality. This slide has summarized the major risk factors we have identified, which may fall into 5 categories. First is demographically. For example, we can see a U-shaped age risk, that is, young children and the elderly are most vulnerable. Besides that, some genetic factors such as HLA-DQA1 and interferon AR2 variants may also increase susceptibility. And the second is the behavior and the socioeconomic factors, including the environmental factors such as cold reasons and air pollution. And the third is some lab indicators. For example, lymphopenia, eosinopenia, and elevated CRP may predict poor prognosis. And the first is comorbidities, like COPD, diabetes, etc. And the fifth is the virus-specific factors such as high viral load and specific subtypes. This slide has explored the potential mechanism behind these risk factors. We propose that epithelial barrier dysfunction is still a central unifying mechanism. Disrupted epithelial barrier by allergen, viruses, pollutants, and chemicals could facilitate pericellular penetration, expose hidden viral receptors, and trigger antimicrobial inflammation. Disrupted epithelial barrier could also trigger TSLP, IL-25, IL-33, ILC2, and Th2 responses, leading to type 2 inflammation. Chronic airway inflammation could exacerbate barrier dysfunction through a positive feedback loop, increase systemic inflammation, and cause severe infection. Type 2 immunity is another core mechanism here. Epithelial damage could trigger the release of TSLP, IL-25, and IL-33, which would cause ILC2 activation and trigger type 2 immunity. The consequences of type 2 immunity could worsen respiratory virus infections in the following 5 aspects. The first, it could reversely exacerbate epithelial barrier damage. And the second, it will cause airway eosinophilia and mucus hypersecretion. The third is the dysregulated ILC2. The first is the IL-13, which may modulate viral entry proteins. And the fifth is the suppression of type 1 immunity. Thank you for your watching.